PhD student at the Owen Institute, and today I'm going to talk about my research, which is basically to find communities in networks. So all of you have come across common networks in your life, like social networks, Facebook, Twitter, and so on. And the problem of community detection becomes important in such networks because you want to find how, for example, rumors spread in networks or physically how epidemics spread in different networks. A similar problem occurs in biology, where we have a protein network, and communities over here are functional modules that perform cellular functions in the body, and finding these can help you understand various diseases. So, how do we tackle this problem of community detection? Although I call my pipeline Super.Complex, it's actually pretty simple. So what you do is you take a network and sample a subgraph, which is basically a bunch of nodes and edges, and test whether this is a community or not by using a machine learning classifier. Now, how do we build this machine learning classifier? I actually employ an AutoML pipeline that runs your data through a whole bunch of preprocessors, a whole bunch of machine learning models, and tells you what is the best one for your data. And so, you can use that model to further test your final solutions. And so, what are the possible challenges with using an AutoML pipeline? You want to run through a whole bunch of models. You want to see which parameters you want to use, which ones you want to cross-validate against, and so you need a lot of compute power. And similarly, moving on to the second step of actually sampling subgraphs, this turns out to be an NP-hard problem, so you need an efficient solution for this. So let's look at an intelligent way to actually sample subgraphs. So assume that this is your network. You start out by actually using the machine learning model itself in the process of sampling a community. So we're kind of going to grow our communities by sampling using the model. So let's start out with these two nodes, IF and as our initial community, and look at all of its neighbors. Let's consider A. So now I'm going to put AFI into my machine learning model and ask, hey, what is the probability that this is a community? And it throws out, hey, 50%. And then I look at the remaining neighbors, G, that's also 50%, H, 50%, but I look at E, it's 8%. So I want to choose this neighbor in my process of growing the community. So I choose FIE, and this process continues on. I look at all the neighbors, and let's say I put it into the model. It says A is the best one, and then again, its neighbors choose the best one, and move on to find the community. So to actually grow the maximum number of communities from your network, you want to do this for every pair of nodes in your network, and so that becomes a huge computational problem. But the good thing is, all of these processes are independent, so you can run a massively parallel framework to find your communities. So that brings me to the results. We actually tested it with a human protein network and employed our pipeline Super.Complex to find protein complexes. And this was entirely done with STAMP2 and Lone Star 5, and we found a whole bunch of complexes that revealed actual biology. So the left complex over here is actually found to be implicated in the pathogenesis of leukemia, and the right one is already known in literature, and is dubbed the control freak of the eukaryotic cell. So that kind of brings me to the end of my talk, and I would like to thank the TACCSTER supercomputers that made this possible, and this is